Today I would like to give a presentation on gene expression profiles and protein-protein interaction network analysis in AIDS patients with HIV-associated encephalitis and dementia. The transcriptional profiles on Affymetrix microarray data was obtained from the National Center of Biotechnology Information Gene Expression Omnibus database, which is based on the Affymetrix human gene annotation data that was assembled during data from public repositories. In total, 4 specimens were available based on the Agilent assay platform. Samples corresponded to AIDS patients with and without pre-mortem history of HIV and dementia. HIV status was confirmed by microscopic observation. RNA of HIV-positive patients was determined by a viral load quantification method using GAG primers. To obtain the differential expressed genes between the different groups of AIDS patients with different race, age, and gender background, publicly available microarray dataset were retrieved from the GEO repository using the Agilent assay microchip technology. Fresh frozen autopsied human brain samples were prepared as fragments for microarray gene expression analysis from frontal cortex dissections, which were validated by QRT-PCR. Prior to differential expressed gene determination, the microarray raw data was tested for quality assessment purposes, comparing it to a synthetic array created by taking probe-wise medians. Quality problems are most apparent either from an MA plot where the lowest mover oscillates a great deal, or if the variability of the log4 change values seems greater than those of these arrays in the dataset. All analyzed microarrays revealed no distorted or aberrant loss lines in the MA plots, which often indicate the absence of the potential quality problems. The median and the interquartile range values appeared on each plot related to a center of the vertical spread of the M values. Subsequently, the data preprocessing was performed to eliminate the effect of background noise and to normalize and summarize expression values for each probe set on the database. The samples have 1,500 overlapping genes with a p-value less than 0.05, and no distinct set of differential expressed genes was detected through a screening of 12,000 genes identifying all samples of AIDS patients with and without HIV-induced dementia. In the present investigation, the majority of the enriched genes were upregulated in the samples from AIDS patients with and without HIV-induced dementia. The differential expressed genes were mostly associated with cellular compartment molecular function, including plasma membrane, non-membrane bound organelle involvement, and nucleotide-nucleoside binding. To gain further insights into the biological pathways, the online biological classification tool incorporated in the DAVID database was used. The main goal of the pathway analysis was to identify the pathways that were significantly enriched in the pathological process represented in the brain samples of AIDS patients. Significant enrichment of those differential expressed genes in multiple characters was observed. The most significantly enriched pathways for differential expressed genes in the samples of AIDS patients with and without HIV-associated dementia were ARB and MAC signaling pathways with high numbers of genes especially involved in the late pathway in particular. The gene ontology terms were significantly overexpressed for differential expressed genes-related biological phenomena such as nucleic acid and cellular metabolic processes. From the results of the CAG pathway enrichment analysis, it is clear that the majority of enriched gene ontology terms of differential expressed genes from the samples of AIDS patients was correlated with the cell signaling pathways and the cytosis-mediated in cell-cell adhesion mechanisms. Some pathways, such as neurotrophin signaling pathway, and the pathway involved in the colorectal cancers suggested that neural death features of HIV-related brain damage and high prevalence of neoplastic tumors of the gastrointestinal tract are also present. Network analysis has been shown to be a powerful tool to understand biological responses in health and disease. In the protein-protein network, the nodes are proteins and the edges are functional interactions. To construct the protein-protein interaction network and maximize the coverage of genome, protein-protein interaction data was obtained from the human interactome as a sample of the genome-scale functional network or human reference network. This reference network represents non-redundant, indirect, and non-loop physical protein-protein binary interaction data obtained from the literature. The network is generic and the same regardless of the tissue genotype and pathological state. To obtain correct and relevant protein-protein interaction, only differentially expressed genes related to AIDS patients were mapped on the reference network and the rest was deleted. The new local network was further refined to remove duplicated edges and self-loops comprising only 900 nodes to reveal highly expressed genes with minimal and maximal p-values. The resulting protein-protein network was not weighted since each protein-protein interaction occurred only once. Since the resulting network was too large to provide more specific and detailed information, it proved necessary to divide this network into subnetworks, each of which represented protein subcomplexes or functional modules. A total of 22 subnetworks were found, among which 6 were detected with the interconnection modes more than 20 and node score more than 2. All clustered subnetworks were varied in size, containing a total of 160 proteins and 700 protein-protein interactions. The clusters were mutually inclusive, indicated that nodes were shared between different individual clusters. In each subnetwork, the score was also determined by a score cutoff to define the number of nodes in the subnetwork. The function of the subnetworks mainly corresponded to immune response, regulation of the cell proliferation, and cellular response to the inflammation. The next degree of each network was calculated with degree-sorted circle layout algorithm, which defined hubs as nodes with a degree value of more than 55. The degree corresponds to a number of nodes connecting all the nodes in the network. A higher value for the degree indicated a high connected network, which was likely to be more robust. Subnetwork 1, 2, and 6 were determined as cytoskeleton and protein folding associated networks containing the highest percentage of hubs of highly expressed genes. On the other hand, subnetwork 3, 4, and 5 were defined to associate with RNA modifications, protein synthesis, and inflammatory processes. In summary, the results indicated that the expression of 1,528 differential expressed genes, which were mainly involved in the immune response, regulation of cell proliferation, cellular response to inflammation, signaling, transduction, and bioreplication cycle. The data also shows that the subnetwork 1, 2, and 6 were determined to contain the maximum number of hubs mainly related to the regulation in cellular metabolism in response to inflammation. The discovery of heat shock proteins that play a vital role in CD4 T cells were found to enhance HIV type-mediated transactivation of the retroviral long-terminal repeats, stimulated single-cell HIV infection. It was also determined that heat shock proteins and fibronectin molecules might play important roles in the mechanism of HIV-associated encephalitis, since they were defined to be as hub nodes with a degree value more than 130. I would like to acknowledge the Department of Anesthesia and Critical Care and also funding BMBF and ZKF and thank you for your attention.